Hi, today we're going to cover Chapter 10, Photosynthesis, our second metabolic pathway that we want to learn about. So just as a reminder, photosynthesis is a process whereby we convert solar energy into chemical energy. And this is really important for the entire living world. So even if you're like, I'm a meatitarian, I don't like plants, well, it turns out that cows eat plants. And so this really is important. It's how we take energy from the sun and then we transform it into chemical bonds. Organisms that are able to do photosynthesis are called autotrophs, or you may have called them producers. These are organisms that can obtain their carbon directly from carbon dioxide. We are not autotrophs. We are actually heterotrophs. Heterotrophs are organisms also known as consumers. We obtain our carbon from organic compounds. So the way that I'm able to incorporate carbon into my body is that I have to ingest an organic compound that already has carbon in it. The specific type of autotroph that we want to talk about in this chapter is a photoautotroph. These are producers that obtain carbon from carbon dioxide because they're autotrophs, but they get their energy from the sunlight. And this can include things such as plants, but it's also all of these other items that are over here. Cyanobacteria, algae, unicellular eukaryotes, purple sulfur bacteria. So we begin to talk about photosynthesis, we need to make sure that we understand orientation. As we look at a plant, photosynthesis occurs in any of the green portions of the plant. So we're going to get lots of photosynthesis in the leaves, but we're not going to have photosynthesis occurring in something like the stem or the roots. As we look at a leaf, if we took a cross-section of it, photosynthesis isn't occurring everywhere within the leaf. There are different layers to that plant organ. And so you can see there's sort of a layer on the outside of the leaf, an area, and then there's a layer in the middle. That middle layer is called the mesophyll. The mesophyll contains our cells where we're going to do most of our photosynthesis. You've looked at mesophyll cells in lab when you looked at the Elodea leaf. So within the mesophyll, you can see those cells where photosynthesis is occurring. Related, importantly, is a structure called the stomata. As we start talking about photosynthesis, we're going to talk about how plants have to breathe in carbon dioxide and get rid of oxygen. Well, they don't have lungs, they don't have a respiratory system, so what they're going to actually use are these stomata. They're little openings in the leaf that can open or shut, and this allows for gas exchange. Zooming in on a mesophyll cell down here, there's one mesophyll cell. You can see that cell wall, then it's full of chloroplasts. And there's our chloroplast again, where you should recognize those parts from before: outer membrane, intermembrane space. A single green structure is a thylakoid, a stack of them is grana, and then they're all floating around in stroma. Please be very careful with the following terms: stroma versus stomata. So stomata are the openings in the leaf that allow for gas exchange, whereas stroma is the liquid that is filling up our chloroplast. Another set of terms that you want to be really careful with are the following. So there's this structure, the chloroplast. The chloroplast is our big organelle where we're doing photosynthesis. And then inside the chloroplast, inside the thylakoid discs, there are little teeny pigment molecules, those little yellow dots that I'm drawing in. These little teeny pigment molecules, these are called chlorophyll. So here we've got to differentiate between chloroplasts and chlorophyll. Rule of thumb is chloroplast is the big organelle, whereas chlorophyll are those little teeny pigment molecules found inside the chloroplast. Here's a lot of what we were just talking about, but now written out better for us. The chloroplast is where we do photosynthesis. The leaf is our major organ of photosynthesis. In there, we're going to find the mesophyll layer. Stomata are openings in the leaves for gas exchange, those openings that we've talked about. The chloroplast is the organelle. It has a pigment inside of it called chloroplast. And then we should be already familiar with what thylakoid, grana, and stroma are. As we look at the overall chemical equation for photosynthesis, we come up with this statement. Photosynthesis is 6 carbon dioxides plus 6 water plus light. Then we do photosynthesis, and what do we get out? We get out a glucose molecule, which is the entire reason to do photosynthesis, and then 6 oxygens as waste. You absolutely should memorize this equation. It's going to be 3 things out and 2 things in, sort of the opposite of what we saw with cellular respiration. Note that the reason that an organism is doing photosynthesis is to create this molecule of glucose. That's the whole reason to do photosynthesis. They're trying to make glucose. Why are they trying to make glucose? Because that organism then wants to take that glucose molecule over to a mitochondria and do cellular respiration. Although the oxygen is a waste product for the plant, it is really important for us. So when scientists were first figuring out this mechanism, one really big question was, where does the oxygen come from? There were 2 schools of thought. Let me explain both and then sort of tell you how we figured out where it came from. So one school of thought was the following. Carbon dioxide is a gas. Oxygen is a gas, and so what must happen is you must remove the carbon from the carbon dioxide, and then that turns it into oxygen. So that was one school of thought. And then there was another school of thought that said, no, that's not right at all. Instead, what's going on is that water has oxygen in it. You do photosynthesis, and then you get leftover oxygen. Again, these are just sort of 2 different ideas. To figure this out, what they did was they took 2 plants and they had them in an enclosed environment. They took the first plant and they poured in radioactive carbon dioxide. Then they waited for a while and they checked to see if the oxygen was radioactive. In the second environment, they had that enclosed plant. They poured in radioactive water. They waited for a while and then they checked the oxygen to see if it was radioactive or not. When they did this particular experiment, what they found out was when the water was radioactive that they gave the plant, then the oxygen was also radioactive. Or in other words, where does the oxygen we breathe come from? The oxygen that we breathe comes from the splitting of water within plants through the process of photosynthesis. On the next slide is a picture from your textbook that sort of diagrams this process. Really, the most important part to focus on is what's going on with the oxygen. So again, big take-home message is the oxygen we breathe comes from the splitting of water. And we can see that right here. The water is what's giving rise to the oxygen. Again, don't worry about the rest of those arrows. The big take-home message is what happens, where do we get the oxygen from? So again, make sure that you know the overall chemical equation for photosynthesis. Photosynthesis is a redox reaction. Remember redox, Leo the lion says grr. It is an endergonic process, however. It's requiring energy input. Delta G is positive. It's not spontaneous. So we have to put in energy from the sun, and then we're able to go ahead and create the glucose molecule. You can see that carbon dioxide right here is becoming reduced. It's gaining electrons, or in this case, you can see it gaining hydrogen ions, whereas water is being oxidized. It's losing those hydrogens as it becomes oxygen. Photosynthesis is a little bit easier than cellular respiration because it's only made up of 2 steps. So when you think of the word photosynthesis, it's trying to already break this down for you. The photo part is part of reactions we call the light reactions. The light reactions happen in the thylakoid, and this is the equation for just what's happening in the light reactions. You take light and water, and you generate oxygen, ATP, and NADPH. When we make the ATP in this first step in the light reactions, we make the ATP through a process known as photophosphorylation. So this isn't substrate level phosphorylation. It's not oxidative phosphorylation. It's a new way of making ATP called photophosphorylation. The second part of photosynthesis is that synthesis element. This is known as the Calvin cycle. The Calvin cycle takes place in the stroma, again, that goopy fluid inside of the chloroplast. And what happens in the Calvin cycle is we take the ATP and the NADPH that we just created from the light reaction. So we take that ATP and NADPH we just made, plus carbon dioxide from the atmosphere, and then we generate a molecule known as glyceraldehyde 3-phosphate. We are going to just abbreviate. We're always going to call it G3P. This is a sugar molecule. This particular slide and all of this information that's on it is very, very important. This should probably be the second thing that you put into your brain as you start thinking about Chapter 10. So first most important thing is to know the overall chemical equation of photosynthesis. Second most important thing that I want you to be able to do is I want you to tell me what are the two reactions that make up photosynthesis, where do they occur, and what are the individual reactions just for those sub stages. So please spend some time with this material. Very important, everything that's on this slide. Here's a visualization of what we were just talking about. So you can see we're looking at our chloroplast right now. In our chloroplast, we're talking about the light reaction. So light and water go into the light reaction. It happens in the thylakoids. Then what happens? We do the light reaction. We make ATP and NADPH, and then oxygen is a waste product for the cell. So this is our light reaction. Light and water go into the thylakoid. We get ATP, NADPH, and oxygen out. Now we go to the Calvin cycle. The Calvin cycle takes place in the stroma. So in the Calvin cycle, we're going to use the ATP and NADPH we just made from the light reactions, plus carbon dioxide from the environment, and we'll generate a sugar molecule known as G3P. Notice that I've started talking about a molecule that sounds similar to something we've talked about before, but is slightly different. That molecule is NADPH. This is another electron carrier. It's similar to NADH. We just see that added initial in it because we're talking about plants now. So NADPH is just another electron box, an electron carrier. Let's begin by talking about the light reactions. In order to do this, we need to understand a little bit about the nature of light. So light is part of the electromagnetic spectrum. It travels in waves, and wavelength determines the type or of light that you're looking at. So we could have red light versus something like blue. Whoa, let's actually make it blue. Something like blue light. And what you can do is you can actually analyze and figure out the difference or the length between the wave crests. And so this is what you guys have been doing in lab as you've measured, maybe perhaps memorized that red light has a wavelength of 700 nanometers, whereas blue light has a wavelength of perhaps 500 nanometers. The type of wavelength determines the type of energy. There is an inverse relationship. If you look down here at the bottom, you can see that things that have a short wavelength, things like the purples, actually have a very high amount of energy, so that's opposite, right? Whereas on the other hand, things like reds that have a really big wavelength, right, 700, that's a big number, they're actually lower in energy quality, so it's an inverse relationship. Light is interesting in that it also behaves as though it has particles. So yes, it travels as waves, but it also behaves as though there are particles. Those particles of light are called photons. So in future slides, we're going to talk about a photon of light hitting a leaf. What we're talking about is a particle of light. The way that light works is there are pigments that can absorb light. Some of the pigments that we're going to talk about today include chlorophyll A, chlorophyll B, and carotenoids. Chlorophyll A is our main photosynthetic pigment. This is the one that's doing the majority of the work for us in photosynthesis. Chlorophyll B and the carotenoids are called accessory pigments. The way that light works is that wavelengths that are not absorbed are reflected or transmitted. So if we think about why is a leaf green, the light comes from the sun and it's every single color of the rainbow. As it hits that leaf, it hits pigments that are in that leaf. So there's pigments down here, things like chlorophyll and carotenoids. Those pigments are able to absorb all of the wavelengths of light except for the green light. And that green light is being bounced back or reflected or transmitted back into our eyes, and that's why it appears green. The spectrophotometer is a fun machine that we've been able to use in lab that allows us to quantify this process. In the spectrophotometer, what we have is a piece of light that gets moved through a prism. And so that breaks it up into its different wavelengths. You then choose a particular color of light to be moved through some sort of slit or device that's going to choose just one wavelength of light, and then you shine that light on some substance. So in this case, up at the top, what we're doing is we're shining green light on a green substance. You then have a detector that is able to see how much of that light is able to make it to through the solution you're looking at. In this case, what we see, this substance is green. It's green because it's absorbing all other colors of light except for green, so the green moves through right well. Or in other words, it's going to have a really high transmittance of green or a low absorption. Note that those two values are always going to be inverse. If you've got a really high transmittance, it will have a low absorbance. Down here we took the same substance, we just shown a different color of light on it. So in this case we showed blue light. In this case the blue light is getting absorbed by the green substance, and so it has a low transmittance or a really high absorbance. Using the spectrophotometer, we can create these things called action spectra, where what we do is we take a pigment, something like chlorophyll, and then we shine light on it. We'll shine 400 nanometers of light, then 405, then 410, 420, and we'll see where does it absorb and where does it not absorb. And we come up with these particular graphs that you see before you. You should know how to read these graphs. You should also understand the relative shape that we get from these various pigment molecules. Let's start with chlorophyll A. That's our main photosynthetic pigment. You can see that chlorophyll A does a really good job of absorbing down here in the purple range. It also does a good job of absorbing over here in the red range. It does not really absorb very well between blue, green, yellow. Again, blue and yellow make green, and so what does this pigment look like? It looks green to us because it's absorbing everything except those blue, green, yellows. Those green colors are bouncing back into our eyes. Chlorophyll B and the carotenoids are our accessory pigments. As we look at these, what they're going to do is they are going to broaden the spectrum for us. So if we look at chlorophyll B, you can see that it's able to absorb a little bit more of that blue light. It's also able to absorb a little bit more of that orange light. And so chlorophyll A with chlorophyll B now broadens the amount of light that we can use within the cell. The carotenoids, what they're doing is they are absorbing excessive light. You may remember from earlier that purple light has a lot of energy associated with it. It's very intense light. And so those lower wavelengths, those purple and blues, you can think of it almost as though they're going to overload the system, right? They have too much energy in them. And so instead of frying the circuitry, what carotenoids do is the carotenoids absorb some of that extra energy down there in those purple and blue wavelengths. Carotenoids, you'll see, do not absorb very well in our yellow, orange, red area, and so carotenoids are going to appear orange. So again, big take-home messages from this. You should know that chlorophyll A is your main accessory pigment. It's absorbing those purples and those reds. Chlorophyll B is able to broaden the spectrum of light used. It's absorbing those blues and those oranges. And then carotenoids, they're absorbing excessive light, especially down in that purple-blue range. Using this, you should hopefully be able to answer this question. So this is an absorption spectra for some unknown pigment molecule. So you're looking at one of those absorption spectra and thinking about where it's absorbing versus where it's not absorbing. What I want you to consider is what color would this pigment have to be? So as we think about that, think again, how does color work? And that'll help you answer the question. Go ahead and give it a shot. What we find out is that this is a pigment that would appear blue. If we think about it, right, why is my shirt blue? It's because it's absorbing everything except for blue, right? And so in this graph, we see something that's absorbing everything except for blue. So again, you should be comfortable reading these absorption spectra. What happens when pigments absorb light is that their electrons get excited. So let's look down at this picture in this corner. When a photon of light comes in, it hits a pigment molecule, and that energy causes an electron to get excited up to a higher energy level. It can't just stay up there indefinitely, so it'll fall back down, giving off heat and fluorescing. Or in other words, if I just gave you an Erlenmeyer flask full of chlorophyll and you shine light on it, what will happen is as that light hits all of those pigment molecules, it'll start to glow it'll fluoresce, and it'll get warm. Now, this is obviously not what happens with trees, right? When the sun rises, our trees don't start glowing red and getting hot. So something sort of different is going on in trees. What we find out is that in plants, chloroplasts are found in these structures called photosystems. You should know the structure of a photosystem and how a photosystem works. That's what we'll go through in the next 2 slides. A photosystem has in its center something called a reaction center complex. This is where there's going to be a primary electron acceptor that can accept the excited electrons, and there'll be a special chlorophyll A molecule. So that's the center bit. Around the outside of the photosystem is going to be something called a light harvesting complex. These are pigment molecules embedded in proteins, and those are going to serve as an antenna, and they're going to funnel the energy of the photons towards the center of the reaction. So here's our photosystem. Let's spend some time here. So in the center, we have our reaction center complex. This entire bit is the reaction center complex. In the reaction center complex down at the bottom, we have a special pair of chlorophyll A molecules. And then up here at the top, we have what's called the primary electron acceptor. So that's all the middle part, the reaction center complex. Around the outside edges, we have the light harvesting complex. So it's these weird purple green part, right? So this is the light harvesting complex. These are proteins, so the purple is a protein, and then embedded in it are a bunch of different pigment molecules. So maybe this is chlorophyll A, maybe this is chlorophyll B, maybe this is a carotenoid, but you've got all these different pigment molecules. How a photosystem works is that when a photon of light comes in, I'm going to erase all this so we can walk it through a little bit clearer. When a photon of light comes into the photosystem, it hits that light harvesting complex, and energy is transferred to one of those pigment molecules. That pigment molecule is then going to pass that energy to the next pigment molecule, to the next pigment molecule, and what we're trying to do is we're trying to funnel all of that energy towards the reaction center. So that energy continues being transferred until it hits that special pair of chlorophyll A molecules. When the energy hits that special pair of chlorophyll A molecules, the energy hits the electrons in that chlorophyll A molecule, and they get excited and they shoot up to a higher energy level. When the electrons get excited up, they then get stuck in this primary electron acceptor. They're not going to fall back down and fluoresce. They're not going to give off heat. So this is the reason that plants are behaving a little bit differently than isolated pigment molecules do. There are actually 2 different types of photosystems. They look very similar. Unfortunately, we named them in the order that we discovered them, not in the order that they work. So I'm sorry, the numbering is a little bit wonky, but work with me here. So photosystem 2 is going to function first. The way you know you're looking at photosystem 2 is that when you look at this particular photosystem, so here's our ugly drawing of a photosystem, right? Sort of hot dog bun look. When we look at that special pair of chlorophyll A molecules in the reaction center, this is going to be a molecule called P680. It's called P680 because this is the light that it absorbs the best. It's able to absorb other light, but that's where it absorbs the best. Photosystem number 1, which works second, again, it's just the way it is best at absorbing a wavelength of 700 nanometers of light, and so it has in it a molecule called P700. That special chlorophyll A molecule is P700. These 2 photosystems work together for the light reaction. So to do the light reaction, we're going to be in the thylakoid, and we are going to run now through the process of the light reaction. There are actually 2 different ways that the light reaction can proceed. One is called the cyclic electron flow. We'll talk about that last. Cyclic electron flow we'll see is really important because it generates extra ATP, but it's not the primary pathway. So we're going to come back to cyclic electron flow. Right now, as we talk about light reaction, I want us to talk first about linear electron flow. This is our primary pathway. It's going to use both photosystem 2 and photosystem 1, and this will make both ATP and NADPH. Just to remind ourselves, if we think about what the light reaction is doing, in the light reaction we're taking light plus water, and then we want to generate three things. We want to generate ATP and NADPH and oxygen. So that's what we're sort of looking at, is this process of where we take the light and the water and we're making this ATP, NADPH, and oxygen. Here we have a diagram of the light reaction, again looking at linear electron flow. So we're going to start over here with our photosystem. This is photosystem 2. It tells us it's photosystem 2, but I would know it was photosystem 2 because you can see that it has P680 in the interior. Also, it's working first, so I know that this is photosystem 2. The photosystem is going to work exactly how he's already set it up. So the first thing that happens is light energy comes in to the light harvesting complex and it hits the pigment molecules. Energy is then transferred from pigment to pigment to pigment, and that energy is shuttled or transferred towards that special chlorophyll A molecule called P680. Once that energy hits P680, electrons get excited from P680 and they go up into our primary electron acceptor. So that's the first part. Again, should be really familiar. It's how we already said a photosystem would work. At this point, we need to talk about P680 for a moment. P680 just lost some electrons. If you lose electrons, you're oxidized. You cannot just have molecules that are oxidized or that have lost electrons. So somehow we have to regain electrons or give back electrons to P680. Here's the solution. Here's what photosystem 2 does. Photosystem 2 is going to take water and break it apart. It's going to release the hydrogens and the oxygens, and it's going to steal those electrons from water and replace it into P680. So we've gotten over the problem. P680 has no longer lost its electrons. They were replaced by the splitting of water. The oxygen at this point is just a waste product. The oxygen is going to be released from the plant cell through those openings called the stomata. All right, so let's continue onwards. Let's just start from the beginning. Light comes into the light harvesting complex and hits a pigment molecule. The energy is transferred from pigment molecule to pigment molecule till it hits P680. When that energy hits P680, those electrons get excited and they get shot up into the primary electron acceptor. We now have a problem that P680 has been oxidized. It's lost electrons. So the solution is now to take water, break it apart, rip the electrons out of water to replace those missing ones in P680, and then the hydrogen ions and oxygens are released. We now want to figure out what's happening to that electron that's stuck up there in the primary electron acceptor. That electron that's stuck up in the primary electron acceptor is going to then go down an electron transport chain. Another way to draw this that might be familiar from chapter 9 is something that looks like this, right? These are these proteins embedded in a membrane, and then down at the bottom, we're going to have ATP synthase. So what happens is these electrons in this primary electron acceptor, they're going to go down this electron transport chain, bloop. As they fall down this electron transport chain, they're poof, giving off energy. Electron pops down, poof, giving off energy. We're using that energy to pump hydrogen ions from one side of the membrane to the other side of the membrane. As that hydrogen ion concentration builds up, those hydrogens will then flow through ATP synthase, generating lots and lots of ATP. So what happens next looks really similar to chapter 9. However, we've got a different name for this process. There is not oxygen down here at the bottom. That's not what's going on. Instead, we'll see in a moment that it's actually a photosystem that's causing these electrons to pull down. So this electron transport chain is making ATP. It is making that ATP through a process called photophosphorylation. Just to think about orientation, because orientation is always important as we look at these. As we look at where there's the low hydrogen ion concentration, this is going to be in the stroma. When we think about where there's the high hydrogen ion concentration, this is going to be in the thylakoid space. All right, so let's walk it from the beginning and see if we can see what's going on. Light comes into photosystem 1, it hits a pigment molecule. Energy is transferred until we hit P680. When that energy hits P680, those electrons are excited up into a primary electron acceptor. P680 is oxidized, it's lost an electron, so we take water and break it apart. The electrons from water go in to fill P680, and the hydrogen and the oxygen are released as byproducts. The electrons that were stuck in the primary electron acceptor now travel down an electron transport chain. As they do so, they generate a bunch of ATP by the process of photophosphorylation. At the end of the electron transport chain is photosystem 1. Let's talk through photosystem 1. It's going to work very similarly to photosystem 2. I know this is photosystem 1 because it's working second. I can also tell it's photosystem 1 because it contains P700. So in photosystem 1, another photon of light has come in to photosystem number 1. As that energy hit a pigment molecule, energy was transferred from pigment to pigment until it hit P700. When the energy hit P700, the electrons were excited, and the electrons shot up into the primary electron acceptor. Once again, we have the same problem as before. P700 has become oxidized. It has lost its electrons. Photosystem 2 has a slightly different strategy for how to fill in those missing electrons. Photosystem 1 is not going to split water to fill in those missing electrons. Instead, photosystem 1 is going to pull the electrons from photosystem 2. This is the reason that the electrons want to move down that electron transport chain, because P700 is so strongly oxidizing, it's pulling those electrons towards itself as it moves. Now that we have electrons up in our primary electron acceptor of photosystem 1, those electrons are going to be placed into a box and they're going to make NADPH. So here we've got our entire process of linear electron flow in the light reaction. In this process, note what went in. We put light in and we put water in. So light and water went in. We had 3 outputs. We generated oxygen, ATP, and NADPH. The reason to do this particular metabolic reaction was to generate that ATP and that NADPH. They were taking the sunlight energy and they were capturing it in usable forms of energy. The oxygen is just a waste product. Here's a diagram that might help you. I'm never going to ask a question about construction workers, but maybe this will just help you as another visual analogy. So we've got 2 photosystems down here. A photon of light comes into photosystem 1 that excites the electron up. That electron goes down an electron transport chain to make ATP. Another photon of light comes into photosystem 2 that excites the electron up, and we put it in a bucket to create NADPH. Again, if this analogy helps, great. If not, don't stress about the construction workers. Related to linear electron flow is another process called cyclic electron flow. Cyclic electron flow is only going to use photosystem number 1. What it does is it produces extra ATP that we need for the Calvin cycle. When we do linear electron flow, we end up with equal numbers of ATP and NADPH. So if I do linear electron flow and I make 4 ATP, I'm making 4 NADH. When I get to the Calvin cycle, though, it turns out that I'm going to need more ATP than I need NADPH to run the Calvin cycle. So there has to be a way for the organism to generate extra ATP, and this is the answer. So in cyclic electron flow, we produce extra ATP needed for the Calvin cycle. No oxygen is released in the step. So here we can see cyclic electron flow. We're focusing in on photosystem 1. What happens? A photon of light comes into the light harvesting complex and hits a pigment molecule. Energy is transferred until we hit our special chlorophyll A molecule. Electrons are excited up and they go into the primary electron acceptor. All of that is how photosystem 2 always works. Nothing's new yet. Here's the little flip that we need to remember. The electrons in photosystem number 1's primary electron acceptor in cyclic electron flow cycle backwards. That's why it's called cyclic electron flow. Those electrons then cycle down and go down the electron transport chain, making bunches of ATP, and they end up right where they started. And then the whole process repeats. Light comes in, we excite the electrons up, they cycle backwards, they go down the electron transport chain, that makes lots of ATP, and they're right back where they started. And so this process, the light's coming in, we're exciting those electrons, they cycle around, and they make the ATP for us. Again, this is really important to have because this produces extra ATP needed for the Calvin cycle. We have a nice moment now to compare some things that we did in Chapter 9 to things that we did in Chapter 10. So in both Chapter 9 and Chapter 10, we saw chemiosmosis. In both Chapter 9 and Chapter 10, we had the electron transport chain that pumped the hydrogens from one side of the membrane to the other side of the membrane, and then it flew through ATP synthase to generate lots and lots of ATP. So both mitochondria and the chloroplast are making ATP. That's important to remember. Chloroplasts are making ATP as well. The way they do this, though, is very different. And when we think about mitochondria, they're taking food energy and turning that into ATP, whereas in the chloroplast, we're taking sunlight energy to convert it into ATP. The process that you make the ATP is also different. It's called oxidative phosphorylation because oxygen is pulling those electrons down in the mitochondria, versus in the chloroplast, this is called photophosphorylation because light is driving the phosphorylation. We noticed that hydrogen ion concentration was high and low in different portions of these organelles. In the mitochondria, it's highest in the intermembrane space and lowest in the matrix. In the chloroplast, it's highest in the thylakoid space, lowest in the stroma. This orientation is really important. You've got a great picture in your textbook that shows this. The dark gray is showing where there's a higher hydrogen ion concentration. The light gray is showing where there's a lower hydrogen ion concentration. So both of them have the electron transport chain, the spinny rotor thing, making ATP, right? That's similar. But the orientation you can see is a little bit different. One way that I remember where the hydrogen ion concentration is highest is that it's in the intermembrane space. That's 2 words. Or it's in the thylakoid space. That's 2 words. The low hydrogen ion concentration is in the matrix, 1 word, or the stroma, 1 word. And maybe that might help you remember this. Again, very important picture. You should feel comfortable understanding this orientation and where hydrogen ion concentration is lowest and highest in the cell, within the organelle. Now let's move to the second part of photosynthesis, the synthesis part. In this part, we're going to learn about the Calvin cycle. This is a cycle, so we're going to start and end with the same molecule. This is where we build sugar by using the ATP and NADPH from the light reaction. We'll need to know what a molecule called RUBP is, how it is involved in the cycle. We'll also need to know about Rubisco and enzyme. There's going to be a carbon fixation step. This is where you bring carbon dioxide in from the environment and then fix it into a molecule of sugar. There will also be a reduction phase where this is where the molecule is gaining electrons. And eventually through this process, we will make a G3P molecule. A G3P molecule is 3 carbons big, so it's going to require us to do the Calvin cycle 3 times to create this molecule. Let's try to diagram out the Calvin cycle. So the Calvin cycle is a cycle, so we're using a circle. The molecule that we're always going to start and end with is a molecule called RUBP. What happens to RUBP is that there is an enzyme called Rubisco. Rubisco is going to bring in carbon dioxide from the atmosphere and combine it with the RUBP molecule. So RUBP is going to come in, plus the carbon dioxide is going to come in, and Rubisco is that enzyme that's drawing the carbon dioxide into this cycle. Once the carbon dioxide has been drawn into the cycle, we then need to add our energy. We are going to add 9 molecules of ATP and 6 molecules of NADPH. You'll note that we need a little bit more ATP than NADPH, but we're covered because we did the cyclic electron flow in addition to the linear electron flow. At this point, after adding the energy into the Calvin cycle, this allows us to fix 1 molecule of carbon. And we're then going to be back to having RUBP. And we do this again. RUBP is going to bind to a carbon dioxide that's brought in by Rubisco. We put in energy, and then we fix another molecule of carbon. We then do this a third time, and we fix a third molecule of carbon. So this process actually requires us to bring in 3 molecules of carbon. We're just going to do those one at a time. This molecule down here that we've created, this 3-carbon molecule, is what we call G3P. So in order to make a G3P molecule, we're actually doing this cycle 3 times, 1 for each carbon. If we wanted to make a glucose molecule, glucose has 6 carbons, so we would need to do this process 1, 2, 3, 4, 5, 6 times. So each time you go through the Calvin cycle, you're able to fix 1 carbon atom. Here's the prettier picture of this process from your textbook. Again, don't worry about the extra detail, focus on what we just diagrammed out previously. So in this process, you start off with an RUBP molecule. We're going to bring in carbon dioxide with the help of an enzyme called Rubisco. We then make a molecule. We're then going to need to put in ATP and NADPH. You can see 9 total ATP, 6 NADPH. And after we have done this process 3 times, we're able to make a G3P molecule. There is a great Science Friday video to watch for this particular chapter. Please look in the videos element within the modules, and you'll be able to learn about plants growing in space and what they're learning about plants as they try to grow in 0G. The last concept that we want to cover in Chapter 10 talks about what happens in places like Texas. What do plants do when they live someplace where it's really hot and dry? So let's imagine that you're a plant. If you're a plant and it's really hot and dry, what's going to happen is on a really hot day in the middle of summer, you're going to close your stomata. That's a good thing because it's going to mean that you won't accidentally lose all of your water by evaporation. But it's bad because if your stomata is closed, you can no longer do gas exchange. So you can't breathe in carbon dioxide, you can't get out the oxygen. So what happens in plants that are called C3 plants, this is sort of our generic type of plant, something called photorespiration occurs. In the Calvin cycle, Rubisco gets confused. There's not carbon dioxide around because the stomata is closed. There's lots of oxygen that's building up in the plant, and Rubisco adds oxygen instead of carbon dioxide to the Calvin cycle. This is going to consume the oxygen and organic fuel. It releases some carbon dioxide, but it's not producing any sugars or ATP. It is limiting the damaging products, however, the light reaction. Photorespiration is not a really great option if you are a plant that lives where it's hot and dry all the time. So plants have evolved 2 adaptations to get around the problem of photorespiration. The first adaptation is called a C4 plant. C4 plants are going to stockpile carbon in one cell and then export that carbon as needed when the stomata are closed and they need those resources. So here you can see it, carbon dioxide is coming into the cell. It's being stored as a 4-carbon sugar called oxaloacetate. That's a good familiar one. And that's just sort of think of it as a stockpile. And so the cell, when the stomata is open and it's cooler, it's bringing in, and it's just stockpiling that carbon. When it gets hot and outside and the stomata close, then that carbon can be exported to a second type of cell to run the Calvin cycle. So even when it's hot, these organisms are still able to do photosynthesis. These are your sugar canes, your corns, and your grasses. Our second adaptation is CAM plants, which stands for chrysolicean acid metabolism. This is a temporal adaptation. Most plants open their stomata during the day and close their stomata at night. These plants do the exact opposite. They're going to open their stomata at night when it is cooler to bring in carbon dioxide. Then during the day when it is hotter, they'll close their stomata and use that carbon dioxide to run the Calvin cycle. You can remember this as the temporal adaptation because if you think about CAM, it's the AM-PM adaptation, and that might help you remember this is the day-night adaptation. This gets us to the end of Chapter 10, so you should know about photosynthesis, the overall chemical equation. You should be able to talk about the light reaction and the Calvin cycle, and then these 2 different plant adaptations. If you have any questions, as always, please reach out to me during online office hours or via email, and we can clarify as needed.